Hello, my name is Priya Alagason, and my project was defining region-specific interactions of the leading-edge protein complex in Saccharomyces cerevisiae. In cells, a cancerous state is induced when committed mammalian cells reprogram or dedifferentiate. Saccharomyces cerevisiae, or Baker's yeast, may provide implications for the identification of conserved pathways involved in cancerous cell division. One possible method for halting cancerous cell growth would be to stop membrane formation in a dividing cell, and a potential way to stop this formation would be via the leading edge complex, a series of proteins consisting of SSP1, ADY3, and YSW1 found in yeast that guides the developing membrane to closure. So the purpose of my study was to characterize interactions of the leading edge protein complex to better understand the process of prospore membrane formation during yeast cell division. In order to identify interactions, each of the three proteins was broken up into different sized fragments: 6 fragments of SSP1, 4 of ADY3, and 19 of YSW1. These fragments were evaluated using the yeast 2 hybrid system, in which the protein fragments were attached to either a DNA binding domain or activation domain, and if the protein fragments interacted, similar to a key fitting into a lock, they would produce the enzyme beta-galactosidase. The amount of enzyme produced is a way to indirectly measure the strength of these interactions. The amount of enzyme produced was measured using both qualitative and quantitative color change assays, in which a substrate was attached to either a blue or yellow pigment and was placed in the presence of the reaction such that the enzyme could consume it. The first set of interactions tested was between SSP1 and ADY3. The strongest binding occurred between the full-length proteins, and second to this interaction, the N+C and ADY3 229-700 amino acid domains displayed the strongest interactions, so these regions are likely to be the binding domains. The next stage was to test for an ADY3 and YSW1 interaction. The proteins did bind, and the strongest binding occurred between the full-length proteins. Strong interactions also occurred between ADY3 fragment 229-700 and the larger fragments of YSW1. ADY3 amino acids 1 to 254 interacted with all 19 fragments, suggesting that it is a general activation domain. To summarize this, the core length of ADY3 is sufficient to bind both YSW1 and SSP1, suggesting that ADY3 plays a central role in the leading edge complex and forms the middle of the complex in relation to the other two proteins. The middle segment of SSP1 is most likely the binding domain. Finally, YSW1 most likely contains two binding domains that interact with ADY3. These findings suggest that ADY3 is most likely to be the best target for halting membrane formation.